The growth of strong, healthy plants relies on many things: good seeds, proper irrigation, smart planting, and good weather. But what we are really excited about is this fascinating, nearly invisible factor that plays a huge role in the life of plants: microbes. Microbes are critical to creating some of our favorite foods and beverages: breads, cheeses, and even beer. They're all dependent on microbes and fermentation. Because bacteria aren't always bad. The same bacteria that help aid our bodies in digestion and maintain our immune system help plants digest nutrients, enhance root growth, and protect them against disease, heat, flooding, and drought. They're fundamental to our health as well as to the health of plants.
For billions of years, microbes and plants have been intimate partners. What may appear to be inert soil in these pots is in fact teeming with microbial life, bacteria, molds, and fungi. In fact, every square inch of dirt contains millions of these organisms. And these microorganisms don't just live alongside plants, they constantly interact, not only through the root system and the leaves, but also inside the plants themselves.
When it comes to finding ways of using microbes in agriculture, Monsanto and Novozymes are absolute leaders in this field. This is why the BioAg Alliance is so exciting. Novozymes has expertise in discovering, developing, and producing microbes. Monsanto, they have fantastic expertise in field testing and a very, very strong commercial arm for selling this segment. The BioAg Alliance is a powerhouse within innovation for the ag sector.
These microbial products will be like probiotics for plants. This groundbreaking research can be explained in 3 steps. First, isolation and identification. Soil is taken from diverse environments like prairie grasslands and productive farmlands, where millions of microorganisms are present in every teaspoon of soil. Researchers isolate the microbes or bacteria that have very specific interactions with the plants by aggressively washing the plant roots. Those microbes' genus, their species, and unique molecular sequence for each of the strains are then identified. Step 2, competition. The microbes from the first step are tested to see if they can compete in a natural environment while also colonizing roots. For example, a microbial pool is created and exposed to plants like corn or soybeans and tested in real soils from the Midwest. This sort of testing, which hasn't been done before, enables enrichment for microorganisms that not only colonize the roots, but also can compete in real-life environments. Microbes that are successful at competing are then produced via fermentation, concentrated, and coated onto seeds. The microorganisms are thoroughly tested, subjected to detailed molecular analysis and reviewed by a safety panel that includes medical experts. This really ensures the safety of our products. The third and final step is field testing. The goal here is to identify if these strains have beneficial effects on plant health, vigor, disease, and insect resistance, which will result in increased yield. Genomic sequencing of these strains allows researchers to understand the difference between both the successful and the non-successful microbes, and then target specific classes of isolated microorganisms for the future.
The benefit of using a microbe in agriculture is that they complement and sometimes even replace other pesticides. They are safe and there are no resistance to them. They help the plant utilize nutrients in a better way. They secure a better yield, thereby helping the farmer grow more with less. This is what we are all looking for in a world where we see population growth and where we see change in eating habits.
With farmers more effective in the field, we're finding smarter ways to feed the planet and provide available, affordable food while protecting the environment.